The overall goal of this procedure is to evaluate the contractility of mesenchymal cells that have undergone in vitro inflammatory cytokine-induced epithelial-mesenchymal transition, or EMT.
This method can facilitate the evaluation of mesenchymal cell function after EMT induced by inflammatory immune responses, such as during idiopathic pulmonary fibrosis or airway remodeling in severe asthma. The main advantages of this technique are that it does not require specific devices and that it demonstrates a high quantitative reproducibility. Demonstrating the procedure will be Hideyuki Takeshima and Kosuke Makita, graduate students from my laboratory.
Begin by washing a culture of A549 human lung epithelial cells with 5 to 10 milliliters of PBS. Next, detach the adherent cells with 2 milliliters of trypsin EDTA at 37 degrees Celsius and 5% carbon dioxide. After 3 minutes, transfer the cell suspension into conical tubes containing cell culture medium and spin down the cells in a centrifuge. Resuspend the pellets in 2 milliliters of cell culture medium, reserving a small aliquot for counting. Then seed 0.5 to 1 × 10⁶ cells in 10 milliliters of medium per 10 cm polystyrene dish and incubate the cell cultures for 24 hours.